My talk's going to be a little bit different. I am a pure clinician. I mean, a pure academic and a clinician. I have no business involvement at all. But I have been involved in a lot of regulatory aspects. I served 2 terms as chairman of the FDA's Cellular Tissue and Gene Therapies Advisory Committee. I was on the bipartisan policy commission that made the recommendations for the Regrow Act. But I've done it not only as somebody who runs a lab, but also as somebody who actually takes care of patients. So I'm also a pediatrician. I was on call. I'm on call tomorrow night, and I was on call before I took the plane down up here the other day. So my perspective on this is going to be a little bit different than what you've heard before. But I think, as you heard, it will echo some of the other speakers before me, but it also is. Am I doing something wrong? No, not at all. Okay. It is very important, I think, for the people in the audience to recognize this. So this is a slide that I've been showing since the early days of the stem cell field. And when we first started wondering, when is it time to take, I should mention that I'm going to be talking about this from a stem cell perspective. Even before it was called regenerative medicine, now it's regenerative medicine. And I'm going to focus a lot on the nervous system since I'm trained as a neuroscientist. But I think you'll be able to see that this is applicable across the board. When is it time to pull the trigger? And I'm not just an Ivy Tower scientist. I'm also a clinician, which means that to make no decision often is to make a decision. So this has stood me in good stead. It's time to pull the trigger for a patient when standard of care is suboptimal or there's no therapy that exists. This is really key. When the biologic data makes sense, when you subject it to critical scrutiny. In other words, the preclinical findings are consistent with our knowledge of the cell's biology and our knowledge of the pathophysiology of the disease process. And if no better proven options are jeopardized. So for example, you don't want something that's going to prevent a patient with a brain tumor from getting radiation therapy, because when all is said and done, that still is the thing that shrinks tumors. It's safe, obviously, but also does no harm. In other words, you don't want to challenge a therapy that challenges the well-being, the daily activity of that patient, his ability to interact with his loved ones, or even to be able to have enough money to live out the last few days of his life. So what I like to say is that even if this is what you're interested in, even if you're purely interested in clinical translation, you really are a translational developmental biologist, and you have to recognize, as you come at this, that stem cells are part of intrinsic developmental programs, and you have to understand what the fundamental developmental biology of the stem cell is, because it has certain biologic imperatives, and if you try to alter those, you will fail. And the regulatory people, like myself, will know that. The purpose of a stem cell is to participate in organogenesis and to maintain homeostasis. For the nervous system, you focus on the neural stem cell, and it doesn't matter how you get there. It doesn't matter whether you've pulled it from a fetus, from an adult, you've gotten them from ES cells or iPS cells. Ultimately, it's the biology of that cell that you need to understand. And what that cell's job is to do is to put together the brain, which means not just neurons, but it means oligodendrocytes and astrocytes in exactly the right ratio. It's also important that its job is to maintain homeostasis in the face of perturbations, which means when you do a transplant into a pathologic host, there's a dynamic, and both elements change, and they change in response to these various arrows, and these are the arrows you must understand. So, for example, one of the arrows is the host talking to the stem cell, and what it says is it will influence its fate. It will exercise a kind of a come-hither signal that we call pathotropism. It will influence its fate decisions, but not as robustly as we used to think. It brings you about halfway into the right ballpark, but it doesn't put you in the right row, and we didn't realize that at first. But there is this kind of crosstalk here. There's a crosstalk between the stem cells once they've actually made it there. They know to have a division of labor, and the division of labor often is that if I'm the neuron, you are the non-neuron, and now there's a communication of this cell talking back onto the host. We used to think it was only diffusible factors, but we know it's not just diffusible factors. We know, for example, that there's cell-cell contact through gap junctions, and more recently we understand that little bits of the cell kind of bud off and will actually bind to the recipient cell and sometimes change it in irreversible ways, kind of, even though it looks like it's a hit and run. Taken together, we started recognizing that there were other actions of the stem cell, and this came to be called the chaperone effect, or other people call it the paracrine effect. This is now about 15 years old. But what it does is it changes the host. Sometimes it can restore function if a lysosomal enzyme is missing, for example, because these are cells that have their normal housekeeping programs ongoing. They spill out lysosomes, and if the host is one that needs it, it will take it up and it'll be restored. But it also neuroprotects, it does anti-inflammatory actions, it inhibits scarring, it will mobilize the endogenous cells there, not only neural cells, for example, and make those send neurites out, but also the other cell types. For example, it promotes angiogenesis. And then, of course, the entire niche will be changed. These all happen not because we lucked out, but because the job of the stem cell is to maintain homeostasis. And we're fortunate, but we have to understand the circumstances. So the biggest question in regenerative medicine is, how do we really best exploit stem cell biology? And I would contend, all these years later, we still do not understand. What we do know is that you cannot make the stem cell do what you think you want it to be doing. I often say that even the dumbest stem cell is smarter than the smartest neurobiologist. Of all of these things, the number one conundrum still remains for most diseases. We actually do not know what needs fixing. Even when we think we know what needs fixing, like in ALS or Parkinson's disease, we don't know what needs fixing. We're wrong more often than we're not. So I like to talk about these as being the aspects of what the stem cell can do. Now notice I've listed them, and I've presented this before at this conference in previous years, but I want to reiterate it. I'm listing them in the order in which I actually think is the lower hanging fruit in the regenerative medicine and stem cell field. Now we all began over here. Notice that's actually at the bottom. That's where I began. And I'm much more enthusiastic about what's going up over here. Now, we talked about protection, and at least in regenerative medicine, this really is where almost everything going on of any success in the regenerative medicine field to date has been that, with very, very few exceptions that I can think of in any organ system using any kind of stem cell. What we like to do is restoring. And I think we're getting better at that, but the biggest challenge that exists, and I'm going to get into my role in the bipartisan commission in a second, is chronic disease, injury of long-standing. Most patients do not come to us that have been freshly injured or in the early stage of their neurodegenerative process. They've been in wheelchairs for years. They're at their end stage of their neurodegenerative process. If you've been symptomatic, it's probably already too late. But nevertheless, that's what everybody wants to embrace. That's what all the companies, that's what every clinician wants to embrace. So this is the third rail of regenerative medicine and is still a challenge. So how do we approach it? Well, one is we know that and I should also mention that every protective therapy that's been successful has always been in the acute or subacute environment, never in chronic or long-standing disease. With no exception that I can think of. Maybe there's one or two. So the notion is, okay, there is something going on. And we have characterized that, and we know this is now looking at proliferation, but this also is a map of what goes on molecularly and metabolically. This is a stroke-like lesion. And in an animal, in a rodent, after a hypoxic ischemic injury, for the first 10 days, there's this robustness of molecular signaling, and it peaks over at this period of time, and then it becomes quiescent again. So maybe we just recreate this environment out here where the window is closed. And that is the way a lot of approaches have started doing it. But it's not going to be enough to simply do what we did in the subacute environment, but do it longer, more enthusiastically, higher dose, more factors. In other words, it's a whole different terrain. It's a whole different landscape, because once you've had a chronic disease, again, this is the spinal cord or the brain, you get neural reorganization and vascular reorganization. You get encephalocele, you get atrophy, muscle wasting, strictures, you get contractures, myelin loss. Things have changed, and for that, you need an entirely new paradigm. And the new paradigm is often going to take multimodal approaches. It's going to take some bioengineering and some cells and some gene therapy and some detours. And it's the multimodal aspect that just drives regulatory people nuts, let alone the companies that may need to be involved with it. So when we were the chair, both as chairing this committee at the FDA, and then also again confronting what would be an expedited pathway, as the Regrow Act would want to do, somebody comes to us and says, this is what I want to do. And I would say that as an academic, and I would say that the other people on my advisory committee are also practicing clinicians or academics. There's only one biotechnology, one industry representative, and he doesn't have a vote on the advisory committee. We didn't look at CMC and scale up and things like that. That was all important, obviously. That's what gets you into reality. But we heavily looked at mechanism of action, proof of concept, and then is any of this have clinical competitiveness? In other words, is it better than standard of care or better than not doing anything at all? Because as enthusiastic as you can be, if you don't address those, there are hidden problems. So I know this may be hard to read. In fact, I think it's impossible to read. Even better. But this is a table. So the Bipartisan Policy Center convened a bunch of us, and we started trying to understand, could we have an expedited pathway by the FDA? So up here, this says level 1, practice of medicine. What that means is things that clinicians do all the time. They don't really need approval. Sometimes they don't even need their own institutional review board approval. It's what a surgeon does. It's deciding, should I use a steroid here or not? Should I use this device or not? That kind of thing. Those don't come under FDA approval. And then, of course, level 3 that's full guns. That's the whole track that you've heard everything about for the last 2 days. What was thinking was, is there another expedited pathway that we can call level 2? Now, this is where I started having a little bit of problems with my colleagues on this commission. I think that there really are, particularly with autologous cells, there is a way to have expedited approval to be able to short-circuit things. Certainly the FDA did it during the AIDS epidemic. They did it during Ebola. They're trying to do it during Zika. What I had problems was, and I don't know if you can see this, let me read it for you, was some of the examples that they wanted to inject into the bill. What it says is autologous bone marrow or cord blood for cerebral palsy, for autism, for heart disease, for all kinds of things that are not approachable by any logic that I could come up with. We don't even know what causes autism. We don't know what causes cerebral palsy, and cerebral palsy is a wastebasket term. So this was something that I had a lot of problems with. And I did my own edits on this, where I made it very narrow. There are things that I think deserve level 2. And actually, the speakers before, Ken talked about some of them. Staying, for example, in a term that's called homologous. You take a cell from the individual and you put it to do exactly in the same spot, doing exactly the same thing, maybe a slightly different spot to do things. So mesenchymal cells for osteogenic indications, or for certain vascular indications, or things like that. That makes an enormous amount of sense, but whenever you go beyond the science, beyond our knowledge of the cell and of the disease process, I still think you need to be there. So I should tell you, I got kicked off the committee. I did. Brock Reeve and I both got kicked off. But how would we approach, I think you do need an expedited pathway. First of all, it's not possible when you're doing autologous therapies to, you can't vet every single cell, but what you can do is you still need to be tied to proof of concept and mechanism of action, but what you can do is validate a process. The way you would do with a drug, certain standards that absolutely must be adhered to, because that will ensure efficacy and safety and purity, and ensure that you are invoking your mechanism of action. And as we do in the clinical world, if it requires something like JACO, which is the Joint Commission that looks at every hospital, but the regenerative medicine equivalent of that should be fine. But what we really need, and Melissa kind of alluded to that, is what we need is knowledge. And sometimes we just need more knowledge. And the knowledge, unfortunately, and this is also tough if you're involved with NIH, is it's not with a product in mind, it's not even with a clear therapy in mind, and sometimes it's not even hypothesis-driven. Sometimes you have to kind of approach a problem even for knowledge's sake, because you need to generate some hypotheses, some new thinking, some new hypotheses. And that's where I come up here talking about prediction. And we're getting better and better at using stem cells to try, unfortunately in a non-hypothesis-driven way, but using it to generate hypotheses, what is really going on in the disease. And I'd say some of the low-hanging fruit in the stem cell field is actually going to be in personalized medicine. This means, for example, not necessarily a treatment for an individual, but trying to understand the omics of that individual, both the functional omics and his molecular omics. What disease is he going to develop? What drug will be best for that particular disease? What is a new target? And we've started doing this at UCSD. I mentioned I'm a pediatrician. I'm specifically, besides being a neurologist, I'm also a neonatologist. We have IRB approval to take this little bit of cord that's chucked for every baby that's born. So wide capture, non-invasive. You can take this bit of cord and you can turn it into iPS cells. So now what you have is not only the molecular omics, but what I like to call the phenomics of every single baby going forward. So it's think of it as a molecular Framingham study, for those that remember what that is. But the key is going to be not only having the omics, but doing constant clinical correlation with these kids as they get older and older and older. In the NICU, who is it who has gut problems and heart problems? As they get a little bit older, who develops autism? Who develops a brain tumor? Who develops diabetes? Who develops a penicillin allergy or a peanut allergy? And then you come up with biomarkers, and the biomarkers give rise to pathways. The pathways give you a mechanism. The mechanism gives you a new target. And then you can start discovering drugs against those targets. It's non-hypothesis driven, but if you do that, hypotheses will fall out. In the aging field, maybe we can do this in the same way. So we have iPS cells from supercentenarians. These are patients who have an intrinsically slow aging process, and then from progeria patients that have an intrinsically rapid aging process, and then patients with normal aging. And what you do is you make iPS cells from them. iPS cells reset the clock, and what we should be able to do is see these guys race, these guys are slow, and these guys should be normal, but we can add progerin to accelerate it over here. It's non-hypothesis driven, but now we should come up with pathways and markers through the genes, the proteins, the metabolome. Are we profiling the right metric if we start doing that? Well, we all want to look, for example, at the transcriptome, but we're starting to learn that that doesn't help us enough. It's the epigenome is more, and now the proteome is probably even more informative, but it's not just whether the protein is there or not. What we're starting to learn is that it's post-translational modification. It's whether it's phosphorylated or not. Protein alone being present or absent doesn't tell us a lot. The abundance of phosphoproteins does tell us a great amount. And that's what one has to start looking at. And this is a map that's a mess because the correlation between the phosphoprotein and the actual abundance of the mRNA was only about a coin toss, about 50-50. And then from the proteome, we're becoming even more sophisticated. We need to look at the metabolome, the connectome, and now the organoid. Well, what that means is recognizing that cells in a monolayer is not the way the human body plan is put together. We're not in monolayers. So Ilya Singech started looking at neural tube closure in a dish because now he can start looking at problems emanating from the spinal cord or from the central nervous system. And in work that's actually supported by SENS, Lina Mastrangelo came, and she's here in the audience, came up with a very, very clever way of looking at cells taken from iPS cells with patients that are normal or have migrational defects, creating the semblance of a neural tube. And while I was talking, what you could see is that these were normal patients, iPS cells from normal patients, neurons. They were migrating in an inside-out kind of pattern, the way we know the brain is put together. And emulating that, these were neurons taken from a patient with smooth brain, lissencephaly, Miller-Dieker syndrome, because lissencephaly is abnormal. And you can notice they don't do that. They don't have the normal inside out. They don't hang on to radial glial cells. And in fact, these patients are called lissencephaly because they have smooth brain because they don't have migrational problems. But now she has a model for looking at that. She can make cerebroids, which have all the layering that you'd expect in the brain, and she can see that as opposed to what you would see, in a normal cerebroid, now it looks like the cerebroid has that same smooth, uncomplicated surface that the patients actually have. Well, now she has a tool for starting to look at what's going on, but also a tool to start looking at environmental factors that could affect migration. Zika comes to mind, CMV comes to mind, other kinds of genetic problems. But once you have that, now you can start doing preventing and intervening. You may not entirely be able to change this, but you could start changing that, because if you have the target, you can start doing high-throughput screening for drugs that change something that's informative. If you're lucky, it's an FDA-approved drug that you can simply repurpose. Or you can begin to understand, you can prevent a problem before it actually manifests itself. So this is a collaboration that I did with Anders Persson and Bill Weiss. This is a mouse, and I'm not going to go into detail, that develops brain tumors. But what we understood was that the neural stem cells seem to have wrong signals. Rather than going out here and making neurons, they are instead pulled over here, or they just don't get the right signals, and they go up here and have inappropriate gliogenesis and give rise to tumors. Well, we can throw the switch back through MAP kinase inhibitors so that neurogenesis is restored. And when you do that, the animals either do not develop tumors, or the tumors shrink, or they don't develop even further. So now what about this notion of preventing a tumor from forming? Getting in and doing things pre-morbidly, pre-malignantly, before the disease. If you're not able to do that, perhaps you can intervene before birth. So this is a conference about aging, but what I'm talking about is preventing the problems of aging early on. So this is a fetal monkey. We could take, now this is a normal fetal monkey, but imagine it's a human that has Huntington's disease. And you take wild-type neural stem cells and you put it into the ventricles, which we could do quite easily. And those cells integrate and mix with the animals, with the monkey, the fetal monkey's own cells, and now participate in corticogenesis right over there. So if these guys are abnormal and these guys are abnormal, this guy's normal. And you can imagine maybe a disease that is lethal now becomes diluted out and is merely a chronic disease or a minor inconvenience, particularly because the exact same thing that happens is we also repopulate the adult neural stem cell population, which plays a role all throughout life. And then, of course, you come to the most controversial aspect of all. And if you could prevent something, and you understood, now you've generated the hypothesis, you know the gene, you know what to do, how far back are you willing to go? And that is exceedingly controversial, and it involves gene editing, which you've heard a lot before. You can edit over here, and more likely than not, it will stay somatic. But as you go back further and further and further here, you start going germline. I don't have the answer for that, but what would you do? Are we willing to do that? This would really be a way of preventing the diseases of aging. You prevent the time bomb from clicking. Now, as Melissa mentioned, there's constant surprises that confront us. And if you stay around long enough working on a project, what started you out on the project isn't how you end the project. And you can either embrace that and go with the flow and adjust to the new knowledge, or you can be crippled by it. So working in ALS with stem cells, we began quite a while ago. And we did it in the days when ALS was simply a motor neuron disease. And the notion was, well, bad motor neuron, put in a good one. Nice and simple, it was going to be a simple disease. And we did that, and we got amazing survival in the SOD1 mouse, the longest ever shown in this mouse model by any technique. But when all was said and done, we didn't do it by replacing motor neurons. In fact, very few. So we had a few choices. We could have done something like that, or we could have embraced what this was the indication. And it turned out that as our sophistication with stem cells developed, the sophistication of what was going on with the disease also developed. And it turned out that, yeah, the motor neurons are sick in ALS, but equally as culpable are the astrocytes in ALS, which make toxic molecules or fail to protect the motor neurons. And what we were doing was making the endogenous stem cells not crank out astrocytes. So we inhibited that. We replaced them with kinder, gentler astrocytes, which now supported the motor neurons, including gray matter oligodendrocytes, which were also neuroprotective. So multiple actions, multiple cell types, again, which drives regulatory people and commercial people nuts, but that's what needed, and that's what worked. So obviously, it's best to protect than to try to recreate what nature did in early development, at least with the nervous system. I don't think we have a clue how to recreate this. Nevertheless, we want to do good things. All of us, especially clinicians, but also scientists, we all get letters like this. There's a little letter from a little girl in Italy. And the line over here that I'll call your attention to is, I wish you could help me be better. But I think that, and we want to do that, and we want to do it as quickly as possible. And I think there are accelerated routes to do that, but to do the accelerated routes requires us, and I thought this was very appropriate for the Olympics, to still keep in mind, if we stay true to the science, that alone will give us insight into what an accelerated route is. And it doesn't mean, and Ken alluded to this too, that it doesn't mean taking shortcuts. It just means following a pathway that the science indicates. So I'm going to end with this, where I began, which is, when do you pull the trigger? When is a therapy really appropriate, at least from the viewpoint of somebody who works scientifically in the field and takes care of patients, has no business involvement whatsoever, is when the standard of care is suboptimal and you got to do something, and when it makes biologic sense and you're not going to prevent the patient from having something that you know works or put him at risk. And the risk is not just physical risk, it's emotional and financial risk as well. So I'll be glad to take any questions. Thanks.